Good day. My name is Mark Eubanks, and today I will be discussing the Lyon hypothesis and X-linked inheritance. The Lyon's hypothesis, simplified, is when there is more than one X chromosome, the other ones are inactivated. This is called X inactivation. In the case of a normal male, where there is one X and one Y chromosome, there is no X inactivation because there is only one. But in the case of a normal female, where there are two X's, one of them is inactivated. X inactivation occurs randomly, but neighboring like X chromosomes will also inactivate, creating a mosaic expression. This means that X-linked inherited traits that are phenotypic express independently of their genetic expression. Cells that have gone through X inactivation are called barred bodies. This is what a normal nuclei of a cell would look like, with the X chromosome material around the center. Whereas in an X inactivated cell in the nuclei, the X chromosome material has been condensed to the cell wall. Now let's look at X-linked traits for calico cats. In black calico cats, the males and females are both going to have the X chromosome with a dominant allele. The orange calicos are going to have the X chromosome with recessive alleles. And there are tortoise shells, which are only female and are heterozygous with the X chromosomes for both the dominant and recessive allele. As we can see in this Punnett square, when crossing either a orange male or black female, or a black male and orange female, the results for both of these are going to be the same, with 50% of them being tortoiseshell female. Now let's look at an example of X inactivation when we cross a black male and a tortoiseshell female. The black male has an X chromosome with a dominant allele, but because there is only one, there is no X inactivation. In the heterozygous tortoiseshell female, the X chromosome with a dominant allele has been inactivated in this case, meaning the X chromosome with a recessive allele carries over, in which the outcome is going to be either a tortoiseshell female or an orange male. Finally, let's build a branch diagram of when we cross an orange female with a recessive short hair trait with a black male with a heterozygous long hair trait. The first thing we need to do is figure out how many gametes are possible, and there are 4. The second and third are to build Punnett squares for the traits of color and hair length. Now let's build our branch diagram. We start off with our possibilities of color, and we branch those off with our 2 possibilities for hair length. And finally we get our combined phenotypic possibilities. For instance, the possibility of being a tortoiseshell female with short hair is 1/4, and we get that starting with a half of a tortoiseshell female times the other half of a short hair trait, and 1/2 times 1/2 is 1/4, and that's how we get this. Finally, let's reiterate by talking about cloning a tortoiseshell calico. While genotypically the clone will be identical to the parent, due to the randomness and the independent expression of X inactivation, it will not be phenotypically identical. All materials referenced comes from Benjamin Pierce's Genetics and Conceptual Approach, and all art was produced by me. Thank you all very much and have a good day.